as well as notes and MCQs. So recorded classes, you can access them anytime, anywhere. Okay, and notes are basically, they are CBME based question and answers, which consists also of MCQs. And there is also, you can enjoy your doing your self-assessment, where it's a fun zone. And MCQs we have from beginner level to expert level. They are standard multiple choice questions, which are usually asked in all the university exams. So you can use the code Dr. Divya 20 for the first free live class. So we have also, we offer you subscription plans. So three plans are there: the basic, standard, and the mentorship. Mentorship is a wholesome program, which we offer you at a special rate of 50,000. So in addition to the pre-recorded classes, live doubt clarification, as well as quick revision, we also give you specialized, it is one-to-one personalized guidance, as well as notes. So you can use the code Dr. Divya 20 for discount on the subscription. So you can book a slot for personalized counseling sessions using the code, as well as, and coming to the internal exam marathon classes, which we have started since April 22nd. It was for a duration, or it is for a duration of 45 days. So here we are doing rapid revision of all important questions, quick revision of the volatile facts, tackling the MCQ questions also when and where as the topic we cover. And this is actually beneficial for everybody, for all the university exam papers, because they are all standardized questions which are asked in all the university exam papers based on the CBME curriculum. So we, with this, I can assure you that your grades would improve, and you will be top-notch grades in the exams. So you can use the code Dr. Divya 20 for the first free live class. So we shall begin with today's class, that is systemic histology revision. So this slide. So here, when this slide is focused, what are we going to see? So we are seeing this. As soon as we see this, we can identify as hyaline cartilage. And here, if we see, so along with the hyaline cartilage, there are also many other structures which are seen there. So this thing which we see here, it is pseudostratified ciliated columnar epithelium. So when we see this and this structure, okay, we have to immediately identify, okay, as trachea. So that is trachea having those structures, okay. So the trachea will have 4 layers. So we have the epithelial layer, then we have the, where here you can see there is the epithelium, okay, epithelium plus lamina, that is the mucosa layer, the submucosa, the cartilaginous layer, and the below layer will be the adventitia layer. So this epithelium, that is the mucosa, it has the epithelium and the underlying lamina propria. So epithelium here is the pseudostratified ciliated columnar epithelium with the goblet cells. So that is the characteristic feature. And here we have some of the seromucous glands, and hyaline cartilage is present. C-shaped hyaline cartilage will be present when we see in the cross structure, right? So here we see it. So these two things. And where the hyaline cartilage is absent, we will see the trachealis muscle. Next slide. So when we see here, okay, so this one here, if you see, they are all flattened cells lining. There's some space here, and you can see it is leading to a structure here. Okay, so, and we also can see some of the bronchioles. Okay, so when we see those structures, we have to identify this as the lung. Okay, so in the lung, what happens? Lung, this space, what we call, okay, this is all the alveoli. Okay, this is the alveolar duct, this is the atrial, this is the respiratory bronchiole. Respiratory bronchiole will lead to the alveolar duct, the alveoli, okay, the atrium and the alveoli. So these are all the alveoli which we are seeing here. In addition to this, in certain fields, we can also see the, this is a bronchial structure, as well as the, we can see the bronchus, where bronchiole and bronchus, how it will differ is bronchioles. Here we see smooth muscles more, but whereas in the bronchus, we see the cartilaginous plates. So that is how it will differ. Okay. So lung, we will identify by the presence of these alveolar ducts and the alveolar sac, which are lined by flattened cells, that is the squamous epithelium. And there is presence of intrapulmonary bronchus and the bronchus. So bronchus means we'll have the cartilage, because bronchus we see from the primary bronchus, tertiary, up to secondary and tertiary level, and the initial portion. Okay, so basically, why it has this cartilaginous plate is basically it will prevent the collapse of the bronchus. Okay, but as we go down towards the bronchiole and all, by the time the air enters there, the whatever the allergens are present, it should not enter beyond. So immediately what happens is the lungs go into these bronchioles, they go into a bronchospasm. They will not let it enter. Okay, so that is why when we are exposed to any allergens or something, we feel a kind of breathlessness. That is because these bronchioles will contract, and they will close the lumen. So that is why they are having smooth muscles, whereas the bronchus, they are having the cartilage. So, and in the bronchus, you will see, okay, so from the pseudostratified epithelium, as we come down, we will see columnar cells, okay, and in the bronchioles, we will see cuboidal cells. This slide. So here, when we are seeing a kind of stratified squamous epithelium structure we are seeing. And in some places, we can see the keratin cover. Some places it is pointed like a mountaineer, and here it is like a mushroom shaped, okay, like a fungus. So when we see that, we have to identify, and below that, we are all seeing the skeletal muscle. Okay, so we are seeing the muscle. This is all the longitudinal section, transverse section of the skeletal muscle. So when we see this, what should come to our mind? It is tongue, because these are papillae. These are filiform and these are fungiform papillae. Okay, so filiform papillae is what? So both the papillae, whether it is fungiform or filiform papillae, they are made up. I mean, the epithelium here is the stratified squamous epithelium. In filiform, what happens? It is covered by a keratin. Okay, it doesn't have taste buds, so it is covered by the keratin. But whereas when we see in the fungiform, it is non-keratinized. This is not covered by keratin. And we see the taste buds on its surface. Okay, so on its surface, this surface, we see the taste buds. So that is the difference between filiform and fungiform. So actually in the tongue, there are 4 types of papillae: filiform, fungiform, circumvallate, and the foliate. Foliate is sparsely seen. Filiform is the most numerous papillae which is seen all over the tongue. Okay, it provides a rough surface. That is why it is covered with keratin. Whereas the fungiform and the circumvallate, they have taste buds. So how they differ is fungiform will have taste buds on its surface, whereas the circumvallate will have this is the circumvallate. So it has the taste buds on its lateral sides, okay, in these sides. Why this is called circumvallate? Circumvallate is located just in front of the sulcus terminalis, where the anterior 2/3 and the posterior 1/3 of the tongue meet. So here around this papillae, there is a depression, okay, all around. This is the sulcus. There is a sulcus present all around that. So that sulcus, we call it as and it is, since it is present all around, we call it as circumvallate. There is this is also called as vellum. Okay, so there is a space all around it. So that is why it is called as circumvallate. And it is circular, if you see here. Okay, so this is the largest papillae in the tongue. So here, how it differs from this thing, fungiform, here the taste buds are on the sides. Okay, so you can see here. And what happens is here, it is also near its base, no? We see these glands. These are all serous glands, which are called as von Ebner's glands. So this von Ebner's glands secretions, it secretes the serous secretion, which will, where the food will mix with its secretion, and it is spread over this taste buds for the perception of the taste. Okay, so that is the characteristic feature there. Okay, so you can see it here. Right. So what is this slide now? Okay, here if we see, we are seeing the epithelium, which is stratified squamous kind of epithelium we are seeing, and we are seeing some glands, and they look like mucous glands. Okay, so and this layer, it is the submucosa layer. So we are seeing the mucous glands here. So when we see these two structures, that is the lining epithelium, stratified squamous, and mucous glands in the submucosa, we should identify it as esophagus. Okay, esophagus. So esophagus is lined by stratified squamous, non-keratinized epithelium. Okay, because esophagus is what? It forms a passage for the food. So it is always prone to wear and tear. So that is why it is provided with the stratified squamous, non-keratinized epithelium. And we have these mucous glands, okay, esophageal glands in the submucosa, basically because they will secrete the mucus, which will provide a lubricating surface for the smooth flow of the bolus, which goes from the mouth to the stomach through this esophagus. Okay, so always GI slides, we see 4 layers. Okay, the epithelium, that is the mucosa, the submucosa, then the muscularis external layer, and the either adventitia or serosa. So adventitia we see when the structure is not covered by peritoneum, like in the esophagus, it is not within the peritoneal cavity, it is not covered by the peritoneum layer. So here we call it as the adventitia. But when it is covered by the peritoneal covering, then we call it as serosa. Okay, but in esophagus, we don't see. So what do we see in the mucosa? So when we understand the structures in the mucosa, we have the epithelium. Okay, the lamina propria, which supports the epithelium. And below that, there will be one more thin layer of muscle, which we call it as the muscularis mucosae or muscularis interna. This muscularis mucosae, it will help in the the contraction of it, it will help in the secretions there. Okay. And the lamina propria will provide the support for the overlying epithelium. So that is why it is present. So these structures, these 3 things make up the mucosa. Below that, we have the submucosa. So submucosa will have the nerve plexus, the connective tissue there. Okay. And in some structures, we see the glands, which are present, like in esophagus, we saw the mucosal glands, right? As well as duodenum also we will see. Okay. And the last layer will be the, third layer will be the muscularis externa, where usually we see inner circular and outer longitudinal. So circular layer will squeeze. So it will initiate the peristalsis, and which one? The longitudinal layer of muscle, when it contracts, it will push. So in that way, both the muscles, they are complementary in their action towards peristalsis. Now this slide, when we see, what are we seeing here? We are seeing the epithelium, and here throughout the epithelium, we are seeing some glands which are eosinophilic and basophilic stained. Okay, so when we see this is stomach. Okay, so this is basically stomach, and stomach, the fundus part of the stomach. Okay, so fundus part of the stomach here, what we see is the gastric pits in the fundus, they are shallow. Okay, so gastric pits are shallow, and they occupy in the entire length of the mucosa, they occupy only 1/4 of it. And it has a lot of gastric glands. All these are gastric glands. So gastric glands will have different types of cells. Okay, parietal cells, oxyntic cells, mucosa neck cells, then enterochromaffin cells. So these are the cells which present in the gastric glands. And these gastric glands, because of the presence of these all different types of cells, it gives kind of a beaded appearance, which you can see here. Okay, this is the beaded appearance it gives. Okay, and next the submucosa. So usually the fundus as well as the body of the stomach, okay, they show similar features. There is submucosa here, where we have the nerve plexus and connective tissue. Then the muscularis layer. So muscularis layer here, muscularis external layer will have 3. Inner oblique, middle circular, and outer longitudinal. Okay, middle circular, outer, this circular and longitudinal we see in rest also. But here the unique thing is the presence of inner oblique layer. Okay, so oblique layer, why it is present? You see in the stomach, what happens? The bolus is mixed with the gastric juices and the enzymes basically to form the chyme, right? So when it has to be mixed, it has to be kind of churned. Okay, so the circular and longitudinal, circular will squeeze like this, longitudinal will push when it contracts. So, but it doesn't churn as such. But when the oblique muscle is present in oblique directions, it's like, you know, in the washing machine, how the clothes will turn in different directions. Same way, this oblique muscle, when it contracts, it will churn the stomach, and that will help in the proper mixing of the bolus with all the gastric juices. Okay. And the last layer here will be the serosa. So when we show the serosa, okay, the serosa when we draw, we will show. It's not like serosa. We need to just show the epithelium there because it is covered by the peritoneum. Okay, you need to after showing the we show the muscle layer. Okay. So, and then after that, the longitudinal muscle layer, all of this, when we show the adventitia, we will show some adipose cells, we'll show blood vessels, okay, the serosa layer, I'm sorry. So serosa layer, we need to show some connective tissue present there. Okay, and last, when we show in adventitia, we just show a line like this. But in serosa layer, when we are saying, we need to show the squamous epithelium lining the structure. Okay, so squamous epithelium should be present there. So then only it is serosa. Right. So the next layer, the next slide here, what we are seeing. Okay, so here when we see the gastric pits are deeper, there are glands here. Okay, so when we see that, this is pylorus. Okay, the gastric pits will be deeper there, lined by the epithelium is lined by columnar epithelium. There are a lot of glands here. Okay, so here the gastric pits will be occupying 2/3 of the mucosa, and there will be presence of these pylori glands in the mucosa. There was gastric glands. These are pylori glands. Pylori glands are against mucus secreting glands. Okay, so because near the pylorus, the that is the chyme has to move to the duodenum. So these help in neutralizing that a lot of acids of the stomach as well. Okay, when the chyme moves from the stomach to the duodenum. So this is the difference. So usually the question is asked as to compare and contrast the stomach fundus and the pylorus. So basically, the main differences lies in the mucosa layer and in the muscularis external layer. So mucosa layer, here the gastric pits are shallow, here the gastric pits are deeper. Okay, we see gastric glands here, we see mucosal glands here. Pylori glands here, that is mucosal and pylori glands. Okay, this is the things. And in the muscularis externa, there we see 3 layers in this part. And in pylorus, we see 2 layers. And circular muscle layer is thick there in pylorus, especially near the pyloric pyloric canal part. No? So the muscular layer the circular layer of the muscle becomes thick because there it forms the sphincter. Next, this slide. Okay, so if we see here, it is lined by columnar, yes, and it has tall projections. These are all the villi. Okay, and villi, they are like the tongue-shaped. Okay, tongue-shaped like villi, long tongue-shaped. And here, down in the submucosa, we are seeing some glands. Okay, so when we see like this, we have to identify it as duodenum. Okay, so duodenum will have this trunk-like villi, which is lined by simple columnar epithelium. And they will be small amount, this microvilli will be present. Basically, they are all for the absorption part, okay, providing a larger surface area. And in the submucosa, there is Brunner's glands. So Brunner's glands are again the mucosal glands which are present in the submucosa, okay. Rest of the layers remain the same, okay. The muscularis externa as well as the serosa layer will have the same structure. So duodenum here you should know that a portion of the duodenum will be covered by the peritoneum, initial portion, rest is retroperitoneal, okay. And this slide, okay, so here we are seeing the foldings like this, and which is lined by tall columnar cells. The columnar cells are very tall, okay. And here we are not seeing the submucosa. Instead, we are seeing the smooth muscles and fiber tissue. Okay, so when we see that, we have to identify it as gallbladder. Okay, so this mucosal folds, they give kind of, when we take a section, in the section, it looks like, gives a honeycomb appearance, we say, okay, for this. So they are lined by tall columnar epithelium cells. And there is absence of submucosa. And instead of that, there is a layer which is called as fibromuscular. That is fiber tissue, fibers as well as muscles together. So in the gallbladder, instead of 4 layers, we see the mucosal layer, the fibromuscular layer, and the last layer will be the serosa or adventitia, based on which portion is covered by the serosa. If we take a section of the fundus part as well as the body part, which is not in relation to liver, that portion it is covered by the serosa. Okay, so the portion that is which is in relation to the gallbladder fossa, that will not be covered by the serosa. So based on that, we will see the adventitia or serosa there. Then this slide. Okay, so here if we see kind of foldings, the villi are folding, the lamina propria is also projected inside. Okay, so these are all the mucosal folds which are formed. In gross appearance, we will see a lot of mucosal folds which form the plica circularis. So what we are seeing is the plica circularis, and this is the villi. Okay, here when we see this villi, they are like leaf-like villi. Okay, this we call them as the leaf-like villi. Here, this is better. So leaf-like villi, where they are lined by the tall columnar epithelium with micro. Same thing will be there. Only thing is the shape of the villi will change, and here we can also see few goblet cells. Okay, rest structure remains the same. Means we have the submucosa layer with connective tissue nerve plexus, the inner circular outer longitudinal layer of smooth muscle, and the serosa layer, they remain the same. Only differences in the villi. Okay, that is the shape of the villi, presence of lacteals. Lacteals are the lymphatics of the intestine. Okay, small intestine. Now here when we see the villi are all decreased, there are more amount of goblet cells are present. And here if we see, there is aggregation of these lymphatic follicles. Okay, so when we see this structure, what should come to our mind? That it is ileum. Okay, so they are small finger-like villi which is lined by simple columnar epithelium, and these aggregation of lymphatic follicles. Okay, what we see here, we should remember that we are seeing that where in the mucosa layer itself. Okay, that is in the lamina propria, which will extend into the submucosa. Okay, it extends from the lamina propria into the submucosa, and this is called as the Peyer's patches. So these two things are very specific for ileum. Rest of the structures, layers remains the same. So small intestine, here when we see the differences, this is tongue-like, leaf-like, and finger-like villi. Okay. Here it is mainly columnar cells, and here we also see the Brunner's glands in the submucosa. Here there is columnar cells along with goblet cells. Lacteals are present more here. Okay. And here there is short finger-like villi, more amount of goblet cells, and the lamina propria, the Peyer's patches are present, which will extend into the submucosa. Now this slide, if you see, it is just the mucosal folds. Okay, unlike here, where there are all villi, okay, projections, it is not there slightly. Okay, this is just the lining epithelium, no villi, and it is more of, if you see, goblet cells. Okay, so when we see this thing, we have to identify it as colon. Okay, so colon, basically here by the time the digested food reaches the colon, most of the nutrients, everything is absorbed. So whatever there it is formed will be the feces will be going. Feces will be present there. Therefore, for the smooth flow or the smooth passage of the feces, we have this more amount of these goblet cells. Okay, so they secrete the mucosa, which will help. And here in the muscular layer, because in the colon, we know that characteristic feature, there are 3 bands of longitudinal muscles, aggregation of these longitudinal muscles, which forms the tinea coli. Okay, so this is what we will see. This is the tinea coli. Next is this slide. So here it is a lumen-like structure. If you see, you can see large amount of the lymphatic tissue here, that is lymphatic follicles are seen, no villi are seen, just mucosal folds little bit. It is suspended. This is the mesoappendix. Okay, so this structure is the appendix. So these things, one is absence of the villi in the mucosa, a mucosa lined by simple columnar only with goblet cells, and lymphatic follicles are present in the submucosa, which will be extending into the lamina propria. Okay, we saw it in ileum. From lamina propria, it was extending into submucosa, but here from the submucosa, it extends. So because of that, what happens? The muscularis mucosae layer, which is present, it is very disrupted here. Okay, because of that extension. Next, this slide. So here what we are seeing a central canal. Then we are seeing cords of cells and anastomosis of those cords of cells, isn't it? So when we see this, what should come to our mind is liver. Okay, so it is central vein. We are seeing the hepatocytes which are arranged. So one is the hepatic lobule. Okay, so basically hepatic lobule we'll see. And at the junction of these hepatic lobules, we'll see the portal triad. Okay, so portal triad is seen. So these cords of cells, these hepatocytes, they are radiating from this, means the arrangement is that way. It looks like as if they are radiating from here, and they will join each other also. Anastomosis will be there. Portal triad with the hepatic artery, portal vein, and the duct, hepatic duct is seen in the portal triad. Okay, so when we see this, we have to identify it as liver. So you have to explain what do you mean by the hepatic lobule? What do we mean by portal lobule? Okay, so here actually the functional unit is not hepatic lobule. Okay, it's just a structural unit there. So what is this slide? So here if we see suddenly, it looks like the serous acini. Yes, it is serous acini. So should we identify it as serous salivary gland? No. Why? Because in addition to that, we have one more structure here, isn't it? So little lighter staining aggregation of cells. So this is called as the islets of Langerhans. So when we see the islets of Langerhans and the serous acini together, we have to identify it as, yes, pancreas. Okay, so pancreas, here the exocrine part is seen as the serous acini. So same thing like how we saw the serous salivary gland, the lobes, lobules within that serous acini are seen. Okay, so we see all the ducts, interlobar, intralobar ducts, okay, intercalated ducts, all of them are seen. The connective tissue is seen in between. In addition to that, the endocrine part is seen as islets of Langerhans. And whenever you show the endocrine part, you have to associate with blood vessels around it, because endocrine glands, as we all know, it doesn't have ducts. So the secretions are poured directly into the blood vessels. Then here, okay, so now this slide, when we see, what are we seeing? We are seeing kind of tubules here. We are seeing this one, right? What is this now? This is the, it looks, and then we see these rays. Okay, here longitudinal loop-like cells are there, structures are there. So when we see this, we have to identify it as kidney. Okay, so we'll see the glomerulus. So here in the kidney, all of us know it has two areas. One is the outer cortex and the inner medulla. So outer cortex will have the renal corpuscles. Okay, that is where we have this glomerulus, the DCT, PCT, and the medullaries. Inner medulla will consist of renal pyramids, loop of Henle, and collecting ducts. So when you draw, you need to show it that way itself. So how will you differentiate DCT and PCT? Okay, PCT will have smaller lumen. It is darkly staining. Okay, it is striated, it shows striations, whereas DCT is lightly staining and it will have a larger lumen. Understand? So that difference should be shown in the diagram. This slide. When we see, the lumen is star-shaped. And if you closely observe the epithelium is transitional epithelium. Okay. And here in the muscle layer, inner longitudinal outer circular will be there. Okay, so this, when we see, we have to identify it as ureter. Okay, it's the star-shaped lumen, which is lined by transitional epithelium. and the muscle coat as inner longitudinal and outer circular. Okay, so it will have the mucosal layer, the muscular layer, and outer this adventitial layer. Now this slide. So this slide again, when we see here, it is looking like stratified epithelium, yes. But if you see the topmost layer of cells, they do not look like squamous. They look a little bit like ballooning cells, isn't it? So when we see that, it is transitional epithelium. And when you see down here, the muscle layer, this is all muscle layer. So muscle layer is little thicker. So when we see these two things, it should be identified as the urinary bladder. So mucosa is lined by transitional epithelium with the absence of submucosa, and the muscle coat is thick. It consists of 3 layers, okay? Inner and outer longitudinal and middle circular. So it will have 3 layers of the muscle. So that is urinary bladder. So yesterday I told you, when we did the histology general revision, that transitional epithelium, what we mean by transition. So when the bladder expands, it looks flattened. The cells look flattened, okay? Otherwise, when it is in a relaxed state, it will be ballooning cells, okay, will be present on the topmost layer. And these cells are covered by the glycoprotein layer, which we call glycocalyx. So it prevents any absorption of any toxic substances. Neither it secretes nor absorbs, okay, anything. And it protects the mucosa against any toxic substances. Next, what is this slide? So we can see sections, and if you see here within, we are seeing kinds of cells which are at different stages, and these cells here are Sertoli cells. Okay, so when we see this, we have to identify it as testis. So there is, the section is cut section of the seminiferous tubules. So outer covering we can see here, albuginea covering. This has the seminiferous tubules. So seminiferous tubules, within it we will have 2 types of cells, that is the Sertoli cells. Sertoli cells and these different that is stages of the development of spermatogenesis, okay? So you need to show that. So Sertoli cells, basically they are supporting and nutrient cells and interstitial cells of Leydig. Interstitial, between the seminiferous tubules, we'll see the interstitial cells of Leydig. Here you can, within this structure, since these are all low power view, you will not be able to identify each structure very specifically, okay, because we study under low power. That is why these slides are placed here. And you need to identify the slide under that low power. Here, when we are seeing, okay, so here we are seeing again many cut sections of the tubules. Inside, we are seeing some structure here, isn't it? And if you see this epithelium, it is lined by pseudostratified epithelium, columnar epithelium. And here you can see one kind of line, isn't it? That line is stereocilia, okay? So tubules here, they are lined. So when we see these things, we have to identify it as epididymis. So tubules are lined by the pseudostratified columnar epithelium having columnar cells with stereocilia. Stereocilia, unlike the cilia, they are not motile. Okay, they are not like, they don't help in the movement at all. Okay, they do not help in the movement of the sperms, but instead they they are basically the non-motile. And in addition to that, this epithelium will have this tall column, which we call it as principal cells, and basal cells are there, which are short. Then you will see the clumps of spermatozoa, which is seen within the tubular lumen. Okay. So this is epididymis. And surrounding that, there will be layer of smooth muscles are present. Okay. Circular layer of smooth muscles are present between that. So basically the contraction of that helps in the squeezing of the secretions forward. Here, again, this epithelium, the lumen, okay, it's a luminous structure, and if you see here, it is lined by again pseudostratified epithelium, okay. So this one, we identify it as vas deferens. Okay, so this also, like ureter will have, like a bladder will have the outer and inner longitudinal and middle circular layer. Okay, so only thing is the lumen here with the pseudostratified columnar epithelium we have to identify. Okay, what is this slide now? Okay, so if you see here, let me put here. So we are seeing this acinus, which is the prostatic acinus. Okay, this last slide is of prostate. So here we'll have the prostatic acini. Within the acini, because as the if this section is from an older person, it will have here prostatic concretions, which were otherwise called as corpora amylacea or amyloid bodies. So as age advances in the acini, we see it more. Okay, so that is the characteristic feature. And each of these acini, they are all covered, surrounded by the smooth muscles or the fibromuscular stroma. So this is prostate gland. Next, when we see here, what are we seeing? You can see there are different stages of, there is a structure, then there is like an interim, there is kind of thing. No? So when we see this, we have to identify it as ovary. So here these follicles are at different stages of development. Okay. So usually we have the primordial follicle where there is the oocyte, which is surrounded by the, so after ovulation, okay, so you know that there is 2 stages of mitosis which is taking place. So in the first, what happens is at birth it is altered. Okay, it is altered at the which phase? The prophase phase. Then it is completed during ovulation, and again it is arrested at metaphase stage, which is completed at fertilization. So only if the fertilization occurs, every that is follicle which is formed will complete its development, that is mitotic division. Okay, otherwise it doesn't. Sorry, meiosis. I said mitotic, it is meiotic division. Okay, the first meiotic and the second. So here the meiotic division will be complete. So here, since we see these different stages, so this is the primordial follicle where we are seeing. Okay, so primordial follicle will have the oocyte, which is covered by a single layer of cells. Then we see the primary oocyte, that is the primary follicle where the cells will multiply. Okay, so you are seeing the primary follicle. This is the primordial, sorry. Here is primordial follicle. This is the primary, there is the secondary follicle. Okay, this is the tertiary follicle. So by the time tertiary follicle stage is reached, what happens is the secondary oocyte, it is pushed to one corner. Okay, and a large interim is formed. So all these things, when we see, we have to identify it as ovary. Okay, we have to identify it as ovary. And here we will see the ovary is lined on the outside by cuboidal epithelium, which is otherwise called as germinal epithelium. Here, germinal epithelium, it is called because of its embryological role in contributing to the formation of the gonads. Okay, otherwise, after birth, it doesn't germinate. It doesn't form any new cells or anything. Okay, so it is just a misnomer after birth. But before that, in the embryological period, it will actually contribute to the formation of the gonad. So that is why it is called as germinal epithelium. Okay, so we see that. So this is the secondary follicle. So once the follicle is, this thing, ovum is released, okay, secondary oocyte is released. Next, we will have the atretic, or if it is that is after the period of ovulation. Okay, we'll also see the corpora hemorrhagica, corpora amylacea. So all these things we need to know about. So here again, now this is the luminal structure. So what are we seeing here? The mucosa is into many folds, isn't it? So when the entire lumen is covered by the mucosa and we see a lot of branching of the mucosa, first thing to come to our mind will be the fallopian tube. So the lumen here, numerous branching mucosal folds, which is lined by ciliated columnar epithelium. So here the cilia is present to help in the movement of the, that is the ovum, okay, which secondary oocyte which is present there, because the fertilization takes place in the ampulla of the, so after fertilization, it has to move right through the fallopian tube. So that is why we have. And the muscular coat here consists of inner circular and outer longitudinal layer of smooth muscles. And outer covering will be the adventitia. Okay. So the characteristic feature is this. Now here, okay, we are seeing two layers here. Basically, here we see some glands. This is the epithelium and the small glands. Okay. And down here we are seeing some muscles. So when we see this, okay, this is actually the slide of uterus in the proliferative phase. So in the proliferative phase, as the name says, it is proliferating, right? It is developing. So because of that, what happens is the endometrium is thin, okay? Thin, lined by the columnar epithelium. And here we have the uterine glands, which are present within the lamina propria. Here, myometrium is very thick. Because the endometrium is thin, relatively, the myometrium looks thicker, okay? And it has many blood vessels. So this is uterus in the proliferative phase. When we see in the secretory phase, okay, this is secretory phase. So suddenly you can see here how the glands are spiraling, okay? So it spiral glands, you can see spiral vessels as well as the glands. So glands also become very tortuous. So when it is in a cut section, it gives the appearance of sawtooth. Okay, this appearance, we call it as the sawtooth appearance of the glands. And the vessels, blood vessels are also spiral vessels are seen here in the lamina propria. And here when we see the endometrium is very thick compared to the myometrium. Okay, so these are the phases. Proliferative phase and the secretory phase. Next, this slide, when we see, what are we seeing here? This is the spinal cord, yes. So how are we identifying the spinal cord? We are seeing the central canal and the central gray matter, head-shaped gray matter, with a slender posterior horn and a bulbous anterior horn, and surrounding outer peripheral white matter. So there is anterior median fissure and posterior median sulcus here, and it is surrounded by the meningeal layers. Okay, so when we see that, we have to identify it as spinal cord. So white matter will have more of myelinated fibers. That is why it appears white, because these myelinated fibers, they reflect light and appears white to the naked eye. Okay, in H&E stain, of course, it takes up the eosin stain. And the gray matter, it contains these neurons and unmyelinated nerve fibers are present there. This is, if you see here, it is looking like many cells are there, that is, neurons are there. And if you observe, there are pyramidal cells. Okay, these are all pyramidal cells. So when we see these pyramidal cells, we have to identify this structure as the cerebral cortex. Okay, so cerebral cortex will have 6 layers. Okay, so molecular layer, outer granular, outer pyramidal, inner granular, inner pyramidal, and pleomorphic. In addition to that, in the inner granular and inner pyramidal layer, okay, we see what we call it as the horizontal fibers of bilarger. Okay, horizontal fibers of bilarger. So those two things are characteristic features. So in the inner pyramidal cell layer, we see large pyramidal cells, which are otherwise called as cells of Betz. Okay, that we see it in the inner pyramidal layer. These are the things which we need to know. So in the, actually the cerebrum will have 4 types of neurons. Okay, so one is cells, that is Golgi cell, the stellate cells, the pyramidal cells, okay, then the cells of Martinotti and horizontal cells of Kajal. So these are the cells which are present. In the molecular layer, we see a few stellate cells and the cells of Martinotti are seen there. In outer granular layer, we see more amount of stellate cells. Outer pyramidal cell layer, we see small, that is medium-sized pyramidal cells. Inner granular layer, we see the horizontal cells of Kajal, and inner pyramidal layer, we will see the pyramidal cells of Betz. Okay, and pleomorphic, as the name says, it's all mixture of cells are present there. Next, this slide. When we see this slide here, when we are seeing, so this is the cortex portion. This is the white matter. Okay, that is the medulla portion. So cortex and the medulla. Cortex will, as she is seeing, if you closely observe, there is 3 layers. This is outer. Then there is one single layer of Purkinje cells. And then there is this layer. Okay, so when we see this Purkinje cells, thin layer, we have to identify this structure as cerebellum. Okay, so cerebellum will have the cortex will have the outer molecular, the middle Purkinje cell layer, which is just single layered, and the inner granular cell layer. Okay, so and it gives like a leaf-like pattern, which is covered by the hair matter. So here in the cerebellum, we see 5 types of neurons. That is the Purkinje cells, the stellate cells, the basket cells, the granule cells, and the Golgi cells. Okay, out of which all are inhibitory except for the granule cells. In the molecular layer, towards the outer part here, outer part we will see these stellate cells, whereas to the inner portion we see the basket cells. Then we have single flash-shaped cells, which is the Purkinje cell layer. And beneath that will be the granular cell layer. So granular cell layer, you need to know a few things here. In the granular cell layer, we see more of granule cells as well as Golgi cells, okay, as well as the climbing and mossy fibers. So here we see formation of rosette, formation of glomerulus. Okay, formation of rosette and formation of glomerulus is seen there. And there is one more terminology which you need to know is cerebellar island. So what do we mean by rosette? Rosette is this climbing fibers, the mossy fibers which come, the tracks which come towards the cortex of the cerebellum. The terminal end of it will be rounded, will be bulged. Okay, so that is called as the rosette. And around this rosette, the axon of the Golgi and the dendrite of the granule, they make synaptic connections, and that is called as the glomerulus. Okay, it is called as the glomerulus. And in this granular cell layer, wherever these granule cells are absent, those areas we call it as the cerebellar islands. Okay, so these are the things which we need to know. And central core of white matter will be there. Okay, this is the optic nerve. Okay, this is the optic nerve. So how are we saying this is an optic nerve? See, we are seeing it covered by the meningeal layers, and in the center, okay, these are all the perimysium, epimysium, and endomysium layers. Sorry, perineurium, epineurium, and endoneurium layers, because it is optic nerve, right? So we have to use neurium. And in the center, there is 2 blood vessels. When we see the epineurium, perineurium, and the endoneurium with the central blood vessels and covering meningeal layers, we have to identify it as optic nerve. Okay, identify it as optic nerve. Then this if you see closely, there is a cleft here. This side there are more of nerve fibers and some supporting cells, it looks like. And here we can see there are chromophil cells and chromophobe cells. And chromophil cells, again, we have the acidophilic and the basophilic cells, which are mixed up. Okay, and there is a central cleft here. And here in this part, there is colloid filled structures. So when we see this, we have to identify this as pituitary gland. So pituitary gland here will have the anterior pituitary and the posterior pituitary. This is the intermediate pituitary with the colloid. Okay, and what cleft we saw was that of the infundibulum. Okay, so these are the things. So anterior pituitary, I already told you, we have chromophil and chromophobe cells. Chromophil cells are acidophilic cells and basophilic cells, which we will see. And between that, we will have the sinusoids. The spaces are known as sinusoids. In the posterior, so usually anterior pituitary secretes the hormones, right? With the stimulation, the feedback mechanism, it secretes the hormones when and when required. Whereas posterior pituitary here, it is full of nerve fibers. Okay, nerve fibers are present. Posterior pituitary, you have to remember, doesn't produce the hormone. It's like an MCQ question, all these things. Okay, so instead of that, what does it do? It will store the hormone. The hormones are produced elsewhere, that is in the hypothalamus. From there, the hormones come to the posterior pituitary, right? So here, this axon terminals, no? because their neuron cell bodies are located in the hypothalamus. So the cell bodies of these axons, which are in the posterior pituitary. So here the terminal ends will form a small swelling lightning structure, okay, which is called as herring bodies. Okay, herring bodies. And these herring bodies will be storing, keeping the hormone for some time till there is a stimulus to release. Okay, and supporting this fibers, there will be these blue dots, what we are seeing here. Okay, they are known as pituocytes. Pituocytes. Pituocytes basically provide support as well as nutrition to this posterior pituitary. The intermediate pituitary ear in humans functionally doesn't have any significance. So here we see the colloid film material, okay, within the intermediate pituitary. Now here also we are seeing this colloid. We are seeing the cells which are lined by cuboidal epithelium, and here some cells. So when we see this, we have to identify this as thyroid gland. Okay, so thyroid gland colloid, depending upon the base of what is that for? Depending upon it is active state, okay, in a secretory that is active or inactive state, the amount will be differing of the colloid. So these follicles, these are thyroid follicles which are lined by the follicular, that is the cuboidal epithelium. And between these thyroid follicles, we will see the parafollicular cells. These parafollicular cells are not only seen between the follicles, it is also seen between the basement membrane as well as the epithelium. Okay, so the basement membrane as well as the cells, we can see the parafollicular cells. Then here we have seen the thyroid, and we are seeing one more part. So, okay, when a structure is associated with thyroid, it is parathyroid. Okay, so parathyroid, sorry, this is not pituitary. It was some wrong this thing. It is parathyroid gland. So parathyroid gland here. Okay, so this is. This is not pituitary, instead it is parathyroid. So here we see basically two types of cells. One is the oxyphil cells and chief cells. Okay, are present. A lot of adipocytes are there. Okay, so here basically these cells, the cells of the parathyroid, they are arranged in a anastomosing cord and clump pattern. Okay. Now here if you see from here, you are seeing some layers of cells which are here inverted. Here we can see them like column. Okay, here we see them like, anastomosing cords. When we see like that, we have to identify it as adrenal gland. Okay, so adrenal gland, again, it will have a cortex and the medulla. So the cortex part is divided into glomerulosa, fasciculata, and reticularis. Inner medulla will consist of groups of cells, okay, which are arranged in kind of reticular pattern, and they are separated by the sinusoids. These are all white thing. What you see here is the sinusoids. So here in the glomerulosa, the cells will be either like acina-shaped or inverted U-shaped. In the fasciculata is the thicker layer where the cells are arranged in the form of cords. So between that, there will be the sinusoidal spaces. And in the innermost part, the cells are arranged in the, that is a reticular pattern. Okay, so that is called as the reticularis. So here, when we see this slide, okay, so we are seeing a thick parallelly running fibers here, which are collagen fibers, and we are seeing the fibroblasts. Here there is the epithelium, stratified epithelium. Here there is cuboidal or squamous epithelium, and we are seeing 5 layers. Okay, so this is basically the cornea. Okay, cornea here, to remember the layers, we remember it as A, B, C, D, E. Okay, the alphabets. So you can remember the alphabets and remember the layers. Okay. So A, B, C, D, E. So A stands for the anterior epithelium. This is the anterior epithelium. B is for the Bowman's membrane. C is for the connective tissue stroma. D is the Descemet's membrane. And E is the, so D is for the Descemet's membrane. Okay. And E is for the endothelium on the inside. Okay. So, right. So, A, the anterior epithelium, okay, it will be like we have the stratified squamous non-keratinized epithelium. Then we have the outer limiting membrane, which is called as the Bowman's membrane. Then we have the corneal stroma. So corneal stroma is made up of collagen fibers and there are fibroblasts. These fibroblasts are also called as the corneal corpuscles or keratocytes. Okay. So corneal corpuscles it's called. So these fibroblasts. So you should remember that. And the inner limiting membrane, which is otherwise called as Descemet's membrane, and the inner endothelium, okay, which is lined by the flattened cells there or cuboidal cells. So this is cornea. Then this slide. Okay, this slide is of retina. Okay, so when we see this, it is retina. So this is made up of 10 layers of cells. So we have the outer pigmented epithelial layer. Okay, then there is layer of rods and cones. Then outer nuclear layer. So these rods and cones, they are basically the axons, which are from the outer nuclear layer, then outer plexiform layer. So here there will be the axons, here there will be the dendrites from this nuclear layer. Okay, the inner nuclear layer. So they will be synapsing here, the dendrites of one layer with the axons of the other. Okay, so and the axons of one with the dendrites of the inside. So like that, they will be synapsing. Then inner plexiform layer, then the ganglion cell layer, nerve fiber layer. So from this ganglion cell layer begins this optic nerve. Okay, so from there with the axons which begin, from there we will have the optic nerve. So when we say plexiform layer basically consists of the dendrites and axons which form synapses. The nuclear layer will have the cell bodies, okay, of those axons and dendrites. So this structure we call this is the retina. With this, we finish today's revision. Thanks for watching Auricle. You can download the app on Google Play and App Store. And for most, so this thing contents, you can follow our social media handles on our, this is YouTube, Instagram, Facebook, as well as Telegram. So until next time,